Frederick County, Maryland, 1979. A highway crew is cutting through a hillside when the earth gives up something it was not supposed to give up. Bones. Dozens of them. No headstones, no names. The site records say only colored burial ground, and a county archaeologist catalogs what's left, including one molar, slightly worn, root still intact, and seals everything in an acid-free box that goes to a shelf in the Smithsonian's National Museum of Natural History, where it sits for 34 years. That tooth is why you're here. Not the history books, not the genealogy websites. That one molar in that one box in a building most people walk past without knowing what it holds. In 2023, a researcher named Edwin Harney, who had trained in David Reich's ancient DNA lab at Harvard before joining 23andMe, extracted genetic material from 27 individuals buried at a place called Catoctin Furnace, an iron forge. Starting in the 1770s, hundreds of enslaved and free African Americans lived and worked there in industrial heat, breathing metal particles, working hours no free person was asked to work. Dozens died. Their names were not recorded. The forge owners' ledgers cataloged them as labor units, a value, a number. And then Harney's team ran their ancient DNA against the 23andMe database, 9.2 million consented participants, and the matches came back. 42,000 living people shared DNA with those unnamed dead. 42,000. Most of them have no idea. Think about what that number means. Before this study, the pre-1870 wall in African American genealogy, the period before the first census that listed Black people by name, was considered effectively impenetrable. Historians accepted it. Genealogists worked around it. It was called the Wall of Invisibility for a reason. One tooth cracked it open. Among those 27 bodies, the DNA confirmed what the records had deliberately erased. They died as families. 5 reconstructed genetic family units in the soil of that hillside. Mothers, children, siblings. The forge owner's ledger called them units of labor. The genome called them something else. If you've ever genuinely wondered where you come from, not the family story, not the passed down version, but the actual biological truth, subscribe. Because what this science is finding right now goes deeper than most people are prepared for. The researchers found something else in that DNA, something about where these people came from before the ship, before the forge, before the name was taken. And the answer didn't go where anyone expected. Writing requires records. Records require someone choosing to keep them. The forge owner kept records. The genome kept different ones. One woman buried at Catoctin Furnace, estimated age 35 to 45 based on skeletal wear, carried mitochondrial haplogroup L3E3b, her maternal lineage unbroken from West Africa, present today in living populations in Senegal, Gambia, and Sierra Leone. The forge owner's ledger calls her one Negro woman, value unknown. Her DNA has a name, a family, and a 15,000-year history. His ledger has a price. The mtDNA haplogroups recovered from that site, predominantly L1, L2, and L3 sublineages, confirm West and West Central African origins consistent with the Senegambian and Angolan slave trade routes. One individual carried L3E3b, matching populations from the Wolof and Mandinka ethnic groups of modern Senegal and Gambia. At Avery's Rest in Delaware, a burial site dated between 1675 and 1725, researchers recovered 3 African Americans. One carried West African haplogroup E1b1a, one carried West Central African Bantu ancestry, and one single person, one life, in one grave on the Delaware frontier, carried both West African and East African ancestry. The full geographic arc of sub-Saharan African population history compressed into a single body. But here's where it gets strange. The L3 lineage that most of these enslaved individuals carried is not merely African. L3 is the haplogroup from which every non-African human being on this planet descends. Every person alive today whose ancestors left Africa, the entire human family outside the continent, traces their lineage through L3. When those workers were brought in chains to Maryland, they carried in their cells the genetic root of every human being who ever lived outside Africa. The standard narrative frames the middle passage as a severing, a cutting off. Ancestry erased, identity extinguished. The DNA tells a different story. The genetic lineages of the enslaved were older, deeper, and more diverse than those of anyone who enslaved them. The erasure was legal. It was administrative. It was enforced by violence and sustained by law. The genome did not comply. And one lineage in particular, the one carried by the majority of African Americans today, turns out to have a history that doesn't begin in West Africa at all. It begins somewhere else, somewhere older. And the genetic signal of it is still in millions of living people right now. People who have no idea they're carrying a piece of a migration that happened long before any slave ship ever left any dock. St. Helena Island, South Carolina, 2019. A community elder named Marquetta Goodwine, known as Queen Quet, is standing before a hearing in Beaufort County. She's fighting to preserve the last unbroken stretch of Sea Island land from development. She has been making this argument for decades. Land rights, cultural survival, community continuity. What she doesn't have yet, but what is coming, is the genomic evidence that will confirm exactly why the land and the community are inseparable. In 2021, a team of researchers from Wake Forest School of Medicine and the University of Pennsylvania, working in direct partnership with the Gullah Geechee Nation, published the most detailed genetic analysis ever conducted on this community. What Kip Zimmerman, Theodore Scher, David Reich, and their collaborators found was not what popular history had prepared anyone to expect. The Gullah retained significantly higher proportions of African ancestry than almost any other African American population in the United States. More African-specific genomic variants. More precise matches to West and West Central African populations. And the reason is geographic. The Sea Islands, physically cut off from the American mainland by tidal marsh, accessible only by water for most of their history, created a biological seal. The families who stayed retained something the families who left did not. Standard population genetics assumes that isolation produces loss, reduced diversity, accumulated error, vulnerability. The Gullah data inverts this completely. Their isolation from the racial mixing patterns of the post-Reconstruction South preserved ancestry signals that most other African American populations lost across 6 generations. And yet, the data said the exact opposite of what everyone assumed about their specific origins. For decades, the dominant narrative, amplified by the film Amistad, by cultural memory, by the communities themselves, held that Sierra Leone was the ancestral home of the Gullah people. The 2021 genetic study tested that directly, and the answer was, partially, possibly, but far more complex than anyone had said. Gullah ancestry traces to multiple West and West Central African populations, not a single source. The Sierra Leone story was built from the historical record, from what was written down, from what was preserved. The genome wasn't bound by those limitations. Here's what that means. The Great Migration, 6 million African Americans leaving the South between 1910 and 1970, a movement that history rightly calls a liberation, also diluted the most precisely preserved African genetic record in North America. The DNA doesn't call it a loss, it simply records the change. What the Gullah communities held onto, by staying, by fighting, by refusing to leave, turns out to have been a genomic archive. One that the rest of American society came very close to paving over. And the thing that was preserved? It's more complicated than Sierra Leone, much more complicated. Because when researchers expanded the reference panel and looked harder at where those ancestry signals actually pointed, they found something the history books hadn't mentioned. Baltimore, 2023. A middle-aged woman opens her 23andMe ancestry composition report. It reads, 74% African, 22% European, 0.8% Indigenous American. Standard. Expected. She's seen this before. The rough shape of what most Black Americans find. And then she notices a line at the bottom. 0.3% Middle Eastern and North African. She assumes it's a glitch. It is not a glitch. Simon Gravel at McGill University, working with colleagues at Stanford, published a study in 2016 that analyzed the genomes of 3,726 African Americans from across the United States. It remains one of the most comprehensive national assessments of African American genomic diversity ever conducted. And what it found was that 82.1% of African American ancestral lineages traced to Africa, 16.7% to Europe, 1.2% to the Americas. Those numbers on their own are not surprising. What is surprising is what happened when they looked at geography. African Americans living in the South carry more African ancestry than those in the North or the West. The difference isn't enormous, but it's consistent and it's traceable. Because it turns out the Great Migration was not random. People with higher European ancestry were statistically more likely to be able to migrate north, not because they chose to more often, but because lighter-skinned individuals faced measurably less violent enforcement of the racial restrictions that governed who could move, where they could stop, where they could sleep, and whether they arrived alive. The Great Migration is in your genome. There are 3 things this study confirmed. The third one is the one that stays with you. The admixture, the mixing of African and European ancestry, happened almost entirely before the Civil War. In the South, under slavery. The Great Migration didn't create new admixture, it distributed the existing admixture across the country, and the genomic fingerprint of where that distribution went tracks exactly with the documented transit routes, the train lines, the highways, the cities where communities landed and stayed. The genome is a map. It remembers the roads even when the roads are gone. Actually, hold on. Before we go further, I want to ask you something. In our last video, we looked at the Dinka of South Sudan, a population whose genetic preservation surprised everyone who studied them because their isolation had held something ancient that nobody expected to find. If that kind of isolation can preserve something extraordinary in East Africa, what do you think is still waiting to be found in the Sea Islands, in the Mississippi Delta, in the communities that no major genomic study has fully sequenced yet? Put your answer in the comments. I'm asking because what comes next suggests the answer is already in the database. It's just been misread. Most African Americans, it turns out, carry a genetic signal that doesn't trace to the population their ancestors were taken from. It traces to a population that moved into West Africa from somewhere else, long before any European ship appeared on the horizon. A migration nobody chose, a history nobody recorded, and a signal that has been sitting in the data, labeled incorrectly, for years. Misreading has consequences. In genomics, those consequences have names. Marquetta Goodwine was born on St. Helena Island. She grew up in the Gullah Geechee community, in a place where the language, the food, the weaving, and the stories retained deep African features that most of America had long since lost. She became the recognized chiefess of the Gullah Geechee Nation, and when the 2021 genetic study was designed, she was not recruited as a subject. She was brought in as a collaborator. That distinction matters more than it might sound. The Zimmerman and Schur study, which included David Reich's team from Harvard, confirmed that Gullah individuals cluster more closely with Sierra Leonean and broader West and West Central African populations than general African American cohorts do. But it also confirmed something that required the Gullah community's own knowledge to interpret correctly, that their genomic preservation is directly tied to geographic continuity. The families who stayed on the islands, generation after generation, held something that migration, even migration toward freedom and better lives, diluted. The genome arrived at a political argument that Queen Quet had already been making for 30 years, without the science. In 2005, she testified before the United Nations Working Group on Indigenous Populations in Geneva. She argued that Gullah Geechee land rights were, in substance, indigenous rights. The response from most American legal institutions was, prove it. The 2021 genomic study, published 16 years later, was the closest thing to proof that science could offer. A 300-year unbroken chain of biological continuity tied to a specific geography. The genome said, this people, this land, this lineage, these are not separable. Everything I just told you, hold on to it, because it's about to mean something completely different. When developers purchase Sea Island property and displace Gullah families, which is happening right now in ongoing real estate transactions with current market values, they are not simply erasing a culture. They are removing a living population from the geographic conditions that preserved their genomic distinctiveness for 300 years. A land deed in this context is a genomic document. The county zoning board in this context is a genomic institution. The 2021 peer-reviewed study made that argument in scientific language. Almost no one has reported it that way. And then the research team expanded the reference panel, added more diverse African populations than the standard datasets contained, and looked again at the Gullah ancestry signals. What they found pointed somewhere the history books had never connected to the Sea Islands. A genetic signal attributed confidently to one West African ethnic group turned out, under a better lens, to point somewhere else entirely. The attribution wasn't a lie, it was a limitation of the tools. Same data, different instrument, different answer. Which raises the question that this entire field is now sitting with. If a better reference panel changes the answer for the Gullah, how many other answers, for how many other people, have been wrong for the same reason? There are things the records mention, and things they do not. The DNA is not bound by either. Central Africa, approximately 1,000 years before the Common Era, a population carrying the Niger-Congo language family, what we now call the Bantu-speaking peoples, begins moving. Not quickly, not in one wave. Over the next 2,000 years, they carry their language, their agriculture, their cattle, and their genetics across the entirety of sub-Saharan Africa. From the Cameroon highlands, east to the Great Lakes, south to the Cape, west along the Atlantic coast. By the time the Portuguese anchor off Angola in 1482, the moment European slave trading begins in earnest on the West African coast, the Bantu expansion is already complete. The people loaded onto those first ships are not simply West Africans. They are the product of one of the largest human migrations in pre-colonial history. They carry a genetic signature that crossed an entire continent before any European vessel appeared on the horizon. That signature is called haplogroup E1b1a, and it is found in roughly 60% of African American males today. The recovery of E1b1a CTS2447 from a burial at Avery's Rest, Delaware, dated to around 1700, confirms that this Bantu-derived lineage arrived in North America in the very first generation of the slave trade on this continent. One of those bodies in the Delaware soil carried a Y chromosome lineage that had already traveled from the Cameroon highlands to the Angolan coast over 2,000 years, crossed the Atlantic under unimaginable conditions, and arrived in colonial America still intact. What they found next changed everything they thought they knew about this. E1b1a is not simply a West African marker. Its oldest ancestral node does not sit in West Africa, it sits in East Africa. The mutation that seeded the entire Bantu expansion predates that expansion by 10,000 years. The haplogroup that most people, including most genomics platforms, label West African, is geographically imprecise at best and meaningfully misleading at worst. It is a lineage that crossed Africa before it crossed the Atlantic, and it arrived in the American colonies already carrying a history that no slave owner, no census taker, and no record keeper knew to document. The conventional narrative places the Atlantic crossing as the origin event of African American identity. The genomics place it as the third major migration, preceded by the Bantu expansion across the African continent, and before that, the original dispersal of modern humans from East Africa 70,000 years ago. What arrived in chains was already the compressed genetic record of two earlier diasporas. The Middle Passage was not a beginning. It was a transit point on a lineage that had already survived everything history could throw at it. And the specific marker that carries that history, the one in 60% of African American males right now, had its origin traced to a date that stops the timeline everyone was using, cold. Wrong is not the same as a lie, but in genomics, the consequences are sometimes indistinguishable. Nashville, Tennessee, 2025. The Alliance for Genomic Discovery, a collaboration anchored at Vanderbilt University Medical Center, publishes initial results from the sequencing of 35,024 individuals. 80% of them are people of African ancestry. It is the largest whole genome sequencing effort ever focused on this population. And buried inside those sequences, present in roughly 3 to 4% of the African American participants, is a genetic variant called TTR V142I. It causes transthyretin amyloid cardiomyopathy, a heart disease. one that can develop silently for years, with symptoms easily attributed to aging before it becomes dangerous. In European populations, V142I is extremely rare. In African Americans, it is one of the most common hereditary cardiac risk factors documented, and in the general public, it is almost entirely unknown. Here's what the Catoctin data adds to that. The same variant, or closely related cardiac markers, appears in individuals from that Maryland burial site. Enslaved people who worked in extreme industrial conditions, breathing metal particulate, sustaining injuries, living under constant physical stress. The cardiac variant that is shortening lives in their descendants 200 years later was already present in their cells when they were forced into that forge. It survived. The records of how they died did not. This isn't just ancient history. This is in living bodies right now. Earlier this year, researchers from Princeton published a paper in Cell that added another dimension. Led by Lu Chen and her colleagues, the study confirmed something that had been a genuine blind spot in human genetics for a decade. African populations, including the ancestors of African Americans, carry traces of Neanderthal DNA. Not through direct Neanderthal contact, through back migration. Non-African populations who had interbred with Neanderthals thousands of years earlier migrated back into Africa, bringing those sequences with them. African Americans, carrying both deep African ancestry and European admixture from the era of slavery, therefore carry Neanderthal sequences through two separate historical pathways, at different frequencies, arriving from different directions, over different timescales. By the time someone's genome arrived at Catoctin Furnace, in the body of an enslaved worker with no recorded name, it had already passed through the Bantu expansion, the Middle Passage, and a back migration from Eurasia that happened before any of this began. 3 geological eras of human history compressed into one body, in one grave, in one Maryland hillside. The health disparity literature has documented for decades that African Americans experience disproportionate rates of cardiac disease, hypertension, and certain cancers. The genomics are now starting to find the specific variants, like V142I, that carry a biological record of what survival under extreme population stress actually looks like. Not a deficit, not an inferiority, a genomic scar from conditions that no reference panel was ever built to find. Named. The word keeps appearing in this story like something unhealed. Named in records that excluded them, named in studies that underrepresented them. Named, finally, by a database that was built, in large part, for someone else. Howard University, Department of Biology, 2022. A researcher opens a genomic database. She is trying to map African American mitochondrial DNA haplotypes to specific ethnic populations in West Africa. She selects the best available reference panel. It contains more than 90% European-derived samples. She closes it, opens a grant application. She has been here before. She will be here again. That is not a personal failing. It is a structural one, and it has been documented. Fatima Jackson, Carter Clinton, and Jennifer Caldwell, researchers at Howard University, North Carolina State University, and Louisiana State University, published a review in 2023 in Frontiers in Genetics that named it clearly. African Americans are disproportionately underrepresented in every single category of genomic testing: diagnostic, clinical predictive, pharmacogenomic, direct-to-consumer, tumor testing. Every category. And the African reference databases that consumer testing platforms use to decode ancestry remain critically underdeveloped relative to the actual diversity they're trying to represent. Here's where it gets stranger still. When a 23andMe customer with Black American ancestry receives a result that reads 73% Nigerian Yoruba, 8% Cameroonian, 5% Ghanaian, that breakdown was generated by an algorithm whose reference panel draws the vast majority of its statistical power from European genomes. The African component runs on a fraction of the diversity that actually exists on the continent. The result is precise enough to look credible. It is not complete enough to be trusted. And here is the detonation at the center of all of this. Every DNA ancestry result a Black American has received from a consumer testing company is an answer produced by an instrument that was primarily calibrated for someone else. The reference panels are not fraudulent, they are underfilled. The difference is the difference between a map with 80% of its roads missing and no map at all, except the person holding it doesn't know which roads are gone. The missing roads are the ones that lead home. The 2021 Gullah study proved this in practice. It was only possible because the researchers specifically recruited Sierra Leonean African reference populations that most commercial platforms don't include. Without those populations in the comparison dataset, the Gullah's distinctive ancestry would have been averaged out, assigned to a generic category, reported as unremarkable. The same genome, the same person. A different answer depending entirely on which lens was used. There is one dataset that could resolve the most important unanswered questions in African American genomics. A comprehensive continent-wide African genomic reference panel with the same depth and geographic specificity that European reference panels already have. Multiple research groups are building it. Multiple projects are advancing it. None have published the version that would change everything. The answers exist. They're in a lab that hasn't finished its work yet. There are things this science has finished, and there are things it cannot, yet. Catoctin Furnace, Maryland, 2023. A woman drives to the state park where the burial ground was found. She's received an email from a genealogist. Her 23andMe results contain short DNA segments that match individuals from the Catoctin study. She is, statistically, one of the 42,000. She stands at the fence at the edge of the excavation site. There is no marker, no plaque, no name. She knows more about the people buried there than anyone who placed them in the ground, more than anyone who owned them, worked them, or recorded them as a monetary value. She knows their family structure. She knows their haplogroups. She knows where on the African continent their maternal lineages trace, roughly, imprecisely, through the limited reference panels currently available. She knows their probable cardiac genetic risk. She knows they died as families. She does not know their names. And the researchers, when they found her in the database, specifically chose not to contact her. The ethical frameworks governing genomic consent had not yet been developed for a finding of this kind. Before we go any further, I want to ask you something real. Before DNA tests existed, before any of this science was possible, how did your family talk about where your ancestors came from? Was there a country, a region, a story that got passed down? Or was that history simply gone, replaced by silence? Put it in the comments. Not as a data point, but because honestly, across this comment section, those answers form a picture that matters. Now, consider what you're carrying. If your family has roots in North America before 1870, in a Black American community anywhere in the South, there is a non-trivial probability that the Catoctin study contains someone related to you. The database has the segment. The consent framework is still being written. The Bantu expansion signal is in your genome. The L3 haplogroup, the root of every human lineage that ever left Africa, is in your genome. The Great Migration is encoded in the length of your chromosomal segments, traceable to the routes your grandparents' grandparents traveled. The V142I cardiac variant may be in your genome. You may not know it. Your doctor may not know it. You are not missing your ancestry. The instrument is missing the resolution. The Alliance for Genomic Discovery will publish. The African Genome Variation Project is expanding. The Gullah Geechee genomic study is continuing. When those reference panels reach the depth they need, and they will, the results will change what every existing ancestry report means. Not metaphorically, literally. The same person, the same DNA, a more complete answer. Subscribe, because when those studies land, we will be here to go through every finding with you in full. The enslaved workers of Catoctin Furnace left 42,000 living relatives and no names. The science has found the relatives. The names are still in the silence.